When a person begins to lose their sense of self to Alzheimer's, it's a visible struggle against a disappearing world. For the families watching, it's a long-term process of grief as memories and cognitive abilities steadily fade. For over 30 years, the prevailing theory focused on these, amyloid beta plaques. They are dark, tangled protein deposits that build up in the brain tissue, and they became the central target for nearly all medical research. The amyloid cascade hypothesis suggested that if we could just clear these plaques, we could stop the disease. But in massive clinical trials, even when new drugs successfully removed the amyloid from human brains, the patient's cognitive decline didn't stop. The plaques were gone, but the neurons were still dying. This forced researchers to look closer at a process called excitotoxicity. A chemical messenger called glutamate was flooding the space around neurons and overstimulating them until they physically collapsed. The receiver for this signal is a protein on the cell surface called the NMDAR. NMDAR stands for N-methyl D-aspartate receptor, and it's a channel that controls the flow of signals into the cell. But here, biology presented a problem. The NMDAR is the exact same channel the brain uses to learn and store new information. If you block it to save the cell from overstimulation, you also block the patient's ability to form a single new memory. This created a scientific impasse. We had identified a mechanism that destroyed neurons, but we couldn't intervene without shutting down the healthy functions of the brain itself. A team of researchers at Heidelberg University looked for a way out of this stalemate by shifting their focus from what the NMDAR was to exactly where it was located. They found that NMDARs located inside the synapse, the tiny gap where two neurons communicate, are essential. When they are activated here, they trigger a survival program that keeps the cell healthy. However, some NMDARs are located outside that synapse. When glutamate levels rise during the progression of Alzheimer's, these extrasynaptic receptors are the ones that become toxic. The breakthrough came when the team realized these outside receptors were not acting alone. They required a specific binding partner to trigger the destruction of the cell. This means the machinery for memory and the machinery for cell death are practically identical. The only difference is the geographical location of the receptor on the neuron's membrane. They identified the partner as the TRPM4 ion channel. Under stress, TRPM4 drifts and locks into the outside NMDAR at a specific connection called the twin-et interface. This triggers a massive, uncontrolled flood of calcium and sodium rushing into the neuron at once. This creates an immediate physical crisis. The neuron's branches, or dendrites, begin to swell and burst, a process known as blebbing, while the cell's energy centers, the mitochondria, are structurally shredded by the ion flood. Beyond the physical damage, the complex sends a signal to the nucleus that actively suppresses the neuron's survival genes, effectively forcing the cell to stop maintaining itself. This discovery identifies the NMDAR/TRPM4 complex as a primary driver of the cell death seen in Alzheimer's. It explains why clearing amyloid plaques wasn't enough. The plaques are a byproduct of a deeper signaling error that had already been triggered. Using this new map of the NMDAR/TRPM4 complex, scientists engineered a potential treatment, a new class of molecules called twin-F interface inhibitors, specifically a compound known as FP802. This approach is different from previous drugs that simply plugged the NMDAR channel, which saved the cell but also stopped the patient from being able to process new information. FP802 acts like a microscopic wedge. It is designed to slide precisely into that twin-F interface, the exact point where the two proteins are connected. By forcing the two proteins apart, the drug breaks the deadly signaling chain. The toxic ion flood stops instantly, yet both the NMDAR and the TRPM4 are left free to go back to their healthy, independent roles in the brain. In mouse models of the disease, this treatment halted cognitive decline and preserved the structural integrity of neurons. Perhaps most surprisingly, by stopping this downstream damage, the drug also reduced the formation of new amyloid plaques. This same NMDAR-TRPM4 interaction has now been found in other neurodegenerative conditions. The same mechanism appears to drive the loss of motor neurons in ALS and the cell destruction in Huntington's disease. By identifying the specific protein bond that triggers cell death, researchers have moved beyond merely cleaning up the biological waste of these diseases. They have demonstrated that it is possible to stop the molecular process that kills neurons while leaving the machinery of memory intact.